Okay. Hi, everyone. My name is Karishma Chhugani, and I'm currently a third-year PhD student in Dr. Serghei Mangul's lab at the USC Alfred E. Mann School of Pharmacy and Pharmaceutical Sciences. And today I'm going to be talking to you guys about benchmarking of machine learning algorithms to predict mortality in sepsis from transcriptomics data. So to give you guys some background, sepsis basically represents a complex, time-sensitive, and life-threatening medical condition, which is a leading cause for ICU mortality worldwide. And currently, sepsis globally presents 50 million cases and 11 million deaths annually. And the US alone spends $38 billion on sepsis-associated costs. And this number could be very well much higher in low and middle income countries, but this is just the data that's reported. There have been multiple definitions for sepsis, starting with sepsis 1 in 1991, all the way until the most recent one, which is sepsis 3 in 2016. And that can be defined as a dysregulated host response to infection, entailing a cascade of physiological and immunological mechanisms, resulting in eventual severe organ failure. So currently, the scoring systems in the ICU, they use SOFA scores, stands for Sequential Organ Failure Assessment. And this is used to evaluate sepsis prognosis and severity. And this is also included in the sepsis 3 definition. The current treatment landscape consists of early identification of pathogen, appropriate antimicrobial therapy, and organs. So what is the problem? Basically, current diagnostic scoring systems and definitions are not able to capture the heterogeneity that is present within these sepsis-infected individuals. If you have sepsis and I have sepsis, our gene expression and all of our immune functions, everything could be very different. And so this heterogeneity makes predicting mortality very challenging because there's no single set of clinical or laboratory parameters that can reliably predict this outcome for all patients. For example, if everybody here got sequenced, that's a lot of information. What do we do with that information? Could we, it'd be really cool to explore, a precision medicine angle, but will that bring something new to the table? So we were really curious to see if transcriptomics data could help be a good predictor of mortality. And currently, individual studies have attempted to predict this from transcriptomics data, but the problem is a lot of their methodologies are vague and not reproducible. So utilizing publicly available transcriptomics data can pose many technical challenges, whether that's batch effect or data variation. People that are generating this data, they are using different library platforms, different experimental protocols, pre-processing techniques, formats, and so this really increases the data cleaning efforts that are required to make sure that our data sets are all consistent. Also, completeness of metadata. This is a big one. Incomplete metadata can lead to reduced sample sizes after filtering. And so we want to make sure that that is consistent. And so essentially, we had to make tough decisions on whether we can include samples or not in the cohort based on what information is available publicly. So what was our approach? So basically, we created the MSIRS cohort, which included over 1,000 patients diagnosed with sepsis across 16 publicly available data sets. We utilized GO2R, GeoQuery, and Lemma to uniformly synchronize the gene expression values. As I mentioned in my previous slide, because they're coming from different platforms, different labs, we need to synchronize those values. We collected metadata files with relevant information, such as outcome, mortality, age, and sex. And then we performed benchmarking of 6 machine learning algorithms to predict mortality in sepsis patients. So these are the 6 ML algorithms we used. And we split our data set into 80% training and 20% testing, which is accompanied by shuffling and stratification by outcome to ensure there are no biases. So in terms of our results, out of the 6 ML algorithms, the support vector machine model showed the best performance in predicting mortality with an AUC ROC value of 0.84. And then the logistic regression model, on the other hand, showed the lowest performance in predicting mortality with an AUC ROC value of 0.78. And one thing that I did want to point out is that just, this is our preliminary analysis and we don't have that many samples right now. We have around 1,000. Which is why we use a lot of classification techniques. But if we should increase our sample size based on getting more metadata and all these other parameters, hopefully we will be able to expand to deep learning approaches. So our best performing model, the support vector machine model, we utilize that for something called SHAP analysis. And the goal of SHAP analysis is to see which features have a heavy influence on your outcome. And outcome in our case is mortality. So we had a total of 1,703 features. So the genes and age and gender. And so we showcase the top 20 features here. So the orange boxes are correlated with negative outcome, which is mortality. And then in the orange boxes, we also have one metadata, which is aging at the top. And then we have 9 features in green boxes, which are associated with positive outcome, which is survival. So we still need to do further network analysis on these genes, but at a very high level, a lot of these genes are associated with cellular metabolism and also immune response. So yeah. So in terms of the significance, we hope that these transcriptomics-based results have the potential to complement clinical severity scores, which can significantly enhance mortality prediction and hopefully empower healthcare professionals to make more timely and tailored treatment decisions. We hope that there's a broader adoption of our methodology in infectious disease research and extending well beyond sepsis as well. So one limitation of our study so far is that there's a lot of limited metadata availability. And so currently our classification only categorizes patients as being infected with sepsis versus not sepsis or not being infected with sepsis. But there's a lot more to the story there. A lot of metadata is lacking with infectious agents. So whether that is bacterial, viral, fungal, what is causing the sepsis essentially. So hopefully we can, collect more of that metadata, whether that's reaching out to the authors of the publications or that is, rechecking the metadata that's available, and hopefully this can better guide antibiotic treatment selection. So future directions, we want to infer cell type composition values from synchronized gene expression data using a tool developed in our lab called GetIt. We want to collect more severity scores, the type of infectious agents, and then we want to increase the sample size and expand the number and types of ML algorithms that we're utilizing. So I guess the ultimate question is what input of data is going to be the best predictor of mortality? Is it going to be gene expression values alone, gene expression and cell type composition, or both of those with clinical severity scores? And that's all I have for today. These are my references. I would like to acknowledge the Mangul lab that I'm part of. And thank you.